What we have in front of us is a hemisphere that has been sectioned in the mid-sagittal plane. And the purpose of this lesson is to focus on the spaces within the brain. You'll recall that all of this beautiful brain formed from the walls of a tube. And the lumen of that tube is still present in the adult form of the brain. It's what we call the ventricular system. And it's in the ventricles that cerebral spinal fluid is produced. And we want to observe those ventricles and follow the flow of cerebral spinal fluid from the ventricles to the space outside of the brain, and then finally back into the bloodstream. So let's begin by looking at the largest of the ventricles. It's called the lateral ventricle, and it's found just on the other side of this membrane. So what I'd like to do is I'd like to gently dissect away that membrane so that you can see the lateral ventricle. And I can do that by just cutting into this space just underneath the corpus callosum. And I'm going to cut a bit of a flap so you can see that there really is a space on the other side of this membrane. So now with that opening made in the septum pellucidum, you can see a large space within the anterior part of the forebrain. So this is called the lateral ventricle, and it extends forward into the frontal lobe. It runs through the parietal lobe, and even extends back into the occipital lobe. And then there is a branch of this space that wraps around into the temporal lobe on the lateral side of the brain. So the lateral ventricle is by far the largest of these ventricular spaces in the brain. Now, much of the cerebral spinal fluid is produced in that lateral ventricle, and it passes from the space within the hemisphere to the third ventricle, which is found right here along the midline of the diencephalon. Now, in order for fluid to get from the lateral ventricle into the third ventricle, it has to pass through a narrow passageway called the foramen of Monroe, or the interventricular foramen. And I'm going to pass this tube through that space to demonstrate that channel. There we go. So now you can see the tip of the tube that has passed through the foramen of Monroe and allows the communication of cerebral spinal fluid from the lateral ventricle to the third ventricle. Now, notice that the lateral ventricles don't directly communicate one to the other. That septum pellucidum that I dissected away is a barrier that keeps the fluid from one ventricle from getting to the next. So here we have our third ventricle, which again is a narrow slit that is bounded on either side by the hypothalamus and the thalamus above. And cerebral spinal fluid flows from this third ventricle down into the fourth ventricle through the smallest of the channels that we have within our brains. And that channel is a narrow space or a narrow aqueduct that runs through the dorsal aspect of the midbrain. And this thin tube that I have is just about the dimensions of that channel. And let's see if I can pass this tube through that channel from the third ventricle down into the fourth ventricle. And indeed, we see this tube emerging now in the fourth ventricle. In fact, the fourth ventricle allows for the escape of cerebral spinal fluid. Cerebral spinal fluid exits the inferior limit of the fourth ventricle, where there is a space between the cerebellum and the medulla. That space is called the foramen of Magendie. There's also another small aperture deep in the lateral recess of the fourth ventricle called the foramen of Luschka. So from that point, cerebral spinal fluid exits the ventricular system and flows around the entire cerebral hemisphere in the space underneath this tissue called the arachnoid. So the tissue that's surrounding the hemisphere here normally contains about 1 millimeter or 2 of cerebrospinal fluid that forms an important physical buffer around the outside of the brain. Finally, this tissue allows for the drainage of cerebrospinal fluid along the dorsal midline. These rough features of the arachnoid right along the dorsal midline here, these are called arachnoid granulations. And these are one-way valves that allow cerebrospinal fluid to leave the subarachnoid space and drain into the superior sagittal sinus and then back into the venous blood. Now, before we conclude this lesson, and while we have this brain in front of us, I want to highlight one somewhat unusual feature of this particular specimen, and it has to do with the cerebellum. Now, I want to show you the cerebellum from what is likely a normal brain in order to compare with this specimen that we have in front of us. So, here we have a cerebellum from an individual who died without neurological complication. And I want you to notice how tight and full are these tiny little branches or folia that form the cortex of the cerebellum. In contrast, this other brain has an unusual amount of space between the folia of the cerebellum. These spaces are indicative of one particular pathology that seems to result in the atrophy of this anterior and medial part of the cerebellum. And that condition is chronic alcohol abuse. So, although I do not know these individuals from whom these brains were obtained, nor do I know their medical history, the appearance of the cerebellum in this specimen is highly indicative of chronic alcoholism compared to what we see in a healthy-looking cerebellum. And this alcohol abuse that I suspect might have afflicted this particular brain is likely to have impaired the coordination of gait and other aspects of locomotion in this individual in life.